good afternoon, good evening, based on where you are. Sorry it took us a while today to connect. We were having some technical issues, but we are all here now. So I'm just gonna check how many people we have. Okay, we have around 30. I think a few people are still connecting. I'm just going to give the little housekeeping instructions one last time. So welcome on our last day of this webinar series. We were really impressed with the interaction, so we hope you found it as enriching as we did. But as always, please note that all of your microphones are muted and your videos as well. If you have any questions during the two Q&As that we will have today, you can raise your hand and we will unmute you, or you can write in the chat or the Q&A. Yeah, that is all from my side, I think. So I let you enjoy this last session, and I'll leave the floor to Marie-Noëlle. You are muted, Marie-Noëlle. Yeah.
Thank you, Alexandra. Thank you so much. Okay, good morning, everyone. Good morning, good afternoon, good evening, wherever you are. We are so happy to have you again today with us. So we will, I just want to introduce this session. Over the past years, access and benefit sharing, especially in relation to digital sequence information, has become a central and sometimes even controversial topic in the global discussion. It raised some fundamental questions about equity, fairness, sharing, and of the benefits arising from the biodiversity of plant genetic resources. So the goal of today's session is really to shed the light on why GSR has become such a hot topic. So without further delay, I would like to give the floor to Dr. Claudio, who will start the series of the presentation. Thank you.
Thank you very much, Marie-Noëlle, and good time of the day to all of you. It's a pleasure to join you in this important webinar series on, of course, molecular characterization and related digital sequence information issue. Today is, of course, the policy session. We'll have a free presentation in sequence. So in this first one is to set the scene and explain a little bit why access and benefit sharing around the issue of digital sequence information has become a very hot and important topic. Please allow me 1 second while I try to put up my presentation. Let's go. So. Okay. So again, my name is Claudio Chiarolla, and I work for the CGIAR Genebanks Accelerator as legal officer. And I will present together with Rouard. Will provide the next important presentation, which will then explain in more detail how DSI has changed the way in which we do research and development in the area of agricultural R&D. And finally, we will look at more specific treaty mechanisms that deal with the rules and governance of digital sequence information. But for now, I'd like to start simply by inviting you to connect, please, if you have a phone, through the QR code or at slido.com through the number that is there. This is just to get to know each other a little bit better. And if you can connect, we'd like to know what is your primary focus or concern when it comes to managing digital sequence information or genetic sequence data. So is that generating data that you want to deposit and make available through publicly available databases such as INSDC? Or is it rather your concern about managing and making data available through your own specialized in-house databases that you might have? Is it both of them? Or is it some other concern, in which case we would invite you perhaps to also type that in the chat in Teams, and we will come back to those considerations in the Q&A after the first two presentations. Okay, I see that some of you are starting to comment. So it's both of the above, of course, managing and making data available for us in-house databases. So that's great. I think we will indeed explain how the governance rules under the multilateral mechanism for the sharing of digital sequence information under the Convention on Biological Diversity, from both perspectives, both from the perspective of those generating data and those making the data available, even if they are from third parties. So we'll make an attempt to cover both aspects with some granularity. Now, if you allow me, see there are still questions coming in, but let's move to the next pool. If you can answer the following question, that is, what do you think should be included in the definition of DSI? So I know very well that you've been discussing DSI related issues for the past 2 weeks. So you are very much acquainted to the technical and scientific aspect of work with DSI. But I'd like you to choose again from this list of options in terms of the scope of the possible policy definition of what should be considered DSI. And we go from a narrow definition that is just DNA and RNA to more expansive definitions that might include proteins and also metabolites and even associated traditional knowledge. Or perhaps you might not know which answer is most appropriate, and you can choose I don't know. And so I can see that there's sort of an equal number between I don't know and a rather expansive definition, DNA, RNA, proteins, and metabolites. So what I would like to point out at this point is that those who have chosen I don't know, it's sort of correct, because as we will see, both under the Convention on Biological Multilateral Mechanism that we will look at in more details in the third presentation, as well as in other treaty contexts, there's no currently set definition from the policy point of view. There's no legal definition of it. And that is because, of course, as technology evolves, there's an impact that this technology has as to the understanding of what DSI might encompass or not encompass. And therefore, this is left somewhat to the interpretation of the different state parties to those conventions to interpret what the exact scope should be. Or in any case, member states decided that a strict legal definition was not necessary to enable the multilateral mechanism for the sharing of benefits from the use of DSI to enable its functioning. So it's not necessary to have a strict definition. However, there's this famous categorization based on Hussein and co-authors that has been used in the context of the negotiation in the Convention on Biological Diversity, which represents, again, these different elements that could or could not be encompassed within the scope of DSI. And it's interesting, in particular, to check that the information is not just the raw sequence information, but it's also importantly the value-added DSI that could include indeed annotation on gene functions, annotations on the level of expression, and then about the protein structures and how protein folds, which is very important, for example, for medical research, where in the past one could take 30, 40 years to identify the structure of a particular protein, and now with the technology that has come to fruition through artificial intelligence and big data science and so on. Of course, it's possible to make those predictions in a much rapid and effective way. So to now turn our attention to indeed the main question that we try to address in this introductory presentation, why it has become such a hot topic. Well, the important and of course outstanding consideration that has to be made relates to the development of technology. So in these slides, you can see that the cost of sequencing has really dropped 1 million-fold in the past several years. So when the first human genome sequence was produced, it took more than 20 years and it costs a few millions. And of course, the technology is now widely accessible. It is relatively inexpensive to sequence and produce genome sequence information for a large number of individuals. So this has really changed the nature of the game and the science that is being produced. The sequencing of genetic resources has led to an enormous amount of information that is being generated and stored every day. Of course, the main database collaboration known as International Nucleotide Sequence Database Collaboration, or INSDC, shares this information on an open and free access basis. Of course, open source databases can be used by researchers and private companies for product development. And this has raised some concerns by parties to the Convention on Biological Diversity that have feared that benefit sharing obligations under the Nagoya Protocol and under the Convention could be bypassed by just using the sequence information without requiring access to the physical materials which are related under the Nagoya Protocol, and therefore would create a loophole so that nobody, in essence, will be bound by obligation to pay them back benefits when there's commercial product being generated. So in this final slide, again, is to recap and put on chronological order a number of factors on how ABS, or digital sequence information, has become a prominent topic in several international fora and related discussions. So firstly, it's because most of the genetic biodiversity has evolved in the global south. And indeed, that is particularly true for crops and livestock. Secondly, there are unequal capacity to take advantage of genetic resources. So developed countries generally have more technology to reap the benefits from the commercial exploitation and for their economic developments. Thirdly, the international access and benefit sharing legislations that have been adopted and came into force into the 1990s and the 2000s with the objectives to redirect benefits derived from the use of genetic resources to the developing countries. Well, those agreements have not really lived up to expectation in many of the cases, with the a redistribution of benefits to a degree and the magnitude that could really support biodiversity conservation in the provider countries. Then, also importantly, of course, the technological advances. We mentioned these in the previous slides, and the next presentation by Rouard will also very much focus on these. But just to anticipate that new generation of sequencing technologies have drastically increased the speed and lowered the cost of generating sequence information. At the same time, genome editing and gene synthesis are creating an unprecedented ways of using DSI as part of discovery research, including in crop and animal breeding, and therefore there's a widespread concern that this DSI could widen instead of closing the technological gap between the North and the global South. notably because many of these innovations can be protected through patents and may not be available on, terms that are, favorable to potential users and beneficiaries in developing countries. And finally, the international conventions and laws that regulate genetic material focus, as I said, on the material, on the genetic resources, and not so much on the informational content of those materials. So that indeed is a loophole of the regulatory system that was set in place in the late 90s and early 2000s. And those agreements were developed on the model that genetic resource users would need to access genetic material that they would introduce into their product development value chains. And so, for example, under the plant treaty system, the current system, payments under the standard material transfer agreements, as you know, are triggered when the access genetic material is physically incorporated into a new plant variety, and then if other requirements are also met. But there's this element of physical incorporation. So it doesn't, of course, those payments do not account at present for example, the use of digital sequence information. On the other hand, under the Nagoya Protocol, access providers can potentially limit access to the data that is derived from genetic materials, but only when they make access and benefit sharing agreements to provide access to the material in the first place. So there they can include terms in those contracts saying if the user can do sequencing or not and under which terms. But in any case, DSI has opened up the possibility that research and development organization can benefit commercially from genetic resource sequence data that is available online through ANSDC, as we mentioned earlier, and through other databases, which is free from benefit sharing obligations. And therefore, this situation can lead to, for example, users being able to identify research leads by just using the sequence data to synthesize new genetic constructs or edit genetic materials they already have, or which they could obtain from sources that are, unregulated, therefore bypassing, once again, the benefit sharing obligations. And all of this has led to a call in the in 2000 from 2010, let's say, from the adoption of the Nagoya Protocol onwards, to close this gap, which is present again in the Nagoya Protocol in all legislation since then, so that new rules be established for the sharing of benefits from digital sequence information. And of course, this call has triggered down, not just in the Convention on Biological Diversity and the Plant Treaty that we will, analyze in more details in the next presentations, but also under other treaties that are out there, such as the UN High Seas Treaty, that is under the Convention on the Law of the Sea, that provides for a full pillar of this treaty is on access to genetic resources from areas beyond national jurisdiction and sharing of benefits from DSI that are derived from those resources. But also in the context of the World Health Organization, there's a pandemic influenza preparedness framework that has elements that talk about benefit sharing and DSI, as well as in the Global Pandemic Agreement, which has under Article 12 a full pillar on the so-called PABS system, which is the ABS system for pathogens, which is still under development. So we won't provide any details on this, but just to let you know what the landscape is of international agreements and treaty mechanisms that deal with digital sequence informations. That's all for now. So thank you so much. And I'd like to now give the floor to Rory for the next presentation. So I'll stop sharing And there will be some time for questions just after Rory's presentation. Thanks.
Okay, thank you, Claudio. Good morning, everyone. Good afternoon, good evening. Depending on where you are, let me just share my screen. Can you see that? Can everyone see that?
Yes, thank you.
Good, good. So thank you, Claudio. You've given everyone an introduction to how DSI has become such a hot topic in the context of access and benefit sharing. I am now going to give you a little more detail on the science and technology behind this. That is how DSI has changed the way we use genetic resources and how these changes have basically messed up earlier concepts of access and benefit sharing, which means it became necessary to revisit the whole issue. Just as a reminder, the concept of access and benefit sharing was introduced by the Convention on Biological Diversity 33 years ago, in 1992. The basic concept being that benefits derived from the use of genetic resources must be fairly and equitably shared. Since 1992, it's been impossible even to talk about accessing and using genetic resources without, at the same time, determining how the benefits will be shared. The Plant Treaty came into force in 2004 with specific ABS rules for plant genetic resources for food and agriculture. And in 2006, it introduced the Standard Material Transfer Agreement, or SMTA. This agreement has to be used for the transfer of all plant genetic resources for food and agriculture under the Plant Treaty's multilateral system. And under this agreement, 0.77% of the sales of a product must be paid into a benefit sharing fund if the product is not available without restriction for further research and breeding. Where product is defined as plant genetic resources for food and agriculture, such as a plant variety that incorporate genetic material received with SMTA and that's ready for commercialization. So the concept is that you obtain material genetic resources with an SMTA, you use it in your program of research and breeding, crossing a female and a male to create offspring in the normal way, hoping to select the best offspring for commercialization as a product. Of course, it's usually more complicated than this. We have very long pedigrees in many of our crop varieties. But even so, the incorporation of genetic material into a final commercial product is a sound, sensible, rational basis for fair and equitable benefit sharing. That's how the Plant Treaty works, and that's how most CBD agreements work. But DSI changes this, and we've identified 3 aspects that have implications for benefit sharing. And if I can boil them down into one-sentence summaries, this is what they are. First, DSI delinks the use of genetic resources for research and their use for development. Second, DSI-related technologies enable in vitro and in silico replication of units of heredity. And third, DSI enables discovery of unrelated genetic resources containing the same sequences. Now, those brief summaries are probably so brief that they're a bit confusing, so don't worry, I'm going to go through each one of these in turn to explain what they mean and why they are important. So on the first point, that DSI delinks use of genetic resources for research and development, here is perhaps the most obvious illustration. It's probably no exaggeration to say that without Arabidopsis, we wouldn't now be using DSI in crop improvement. Genetic resources of Arabidopsis have been used by the research community to generate the basic knowledge that we now rely on. The value created through research on Arabidopsis is huge, yet none of those genetic resources has been used to create commercial varieties. So benefit sharing based on incorporation of genetic resources into varieties cannot be fair and equitable because it ignores all that value creation with Arabidopsis. It's a similar story with reference genomes of crops, starting in 2002 with the genome sequence of the rice variety Nipanbari. This has been a key component of transforming knowledge about Arabidopsis into knowledge that can be applied to rice. CGIAR got involved soon after the initial reference genome was published in 2002, with the publication in 2014 of over 3,000 whole genome sequence. And as of 2020, we now have 16 so-called platinum standard reference genomes that span the full range of the genetic diversity of Ruaraidh sativa. And as our knowledge has developed, CGIAR has also engaged in developing reference genome sequences for many of the other crops in its mandates, transforming breeding for these crops as well. It's still very expensive and time-consuming to do this work, and you only need a few reference genomes per crop, so only a very small number of accessions have been sequenced like this. But the value it creates is huge, so the same point applies. Although these genomes are enabling the creation of huge value, many of them have been used very little, if at all, in actual crop improvement. So again, benefit sharing based on incorporation of genetic resources into varieties ignores this value and cannot be fair and equitable. If we put this into a diagram, so we have genetic resources used for research, almost all sequence data and most genotyping data, and a large and increasing proportion of genetic material is used in the research phase to understand the dependence of phenotype on genotype. So I've already talked about Arabidopsis and reference genomes. Let's now add genome-wide association studies that many of you will probably have been involved with. That can involve hundreds or preferably thousands of samples from different countries. The outcome of all this research is the knowledge and tools that are needed for more efficient and effective product development. With this knowledge and these tools, you can focus on a small set of genetic resources and a small part of their genomes for actual crop improvement based on research on large numbers of genetic resources that never get included in crop improvement programs. This is a really serious problem for bilateral benefit sharing based on the incorporation of genetic material into products. It's arguably not such a big problem for the plant treaty, because we already have a multilateral system which shares benefits among all member countries. Yes, it's only a few genetic resources that trigger the obligation to make payments, but those payments get paid into a benefit sharing fund which gets distributed to potentially all, regardless of whether their genetic resources have been used for research and development. So we can argue it's only a problem for the treaty where the knowledge and tools are monetized other than through developing material products. For example, through selling access to data. Although even here, it could be argued that those non-material products have value only to the extent that they help develop material products. So possibly the treaty, but not the CBD, already has the basis for sensible benefit sharing with DSI. Then the second point, in vitro in silico replication of units of heredity. Here I'm referring to genetic modification and gene editing, which enable in vitro replication of DNA. And with synthetic biology, we can go a step further, replicating units of heredity on the computer disks before recreating DNA and incorporating them into material products. Actually, when you reflect on this, it may not be such a big problem because neither the CBD nor the treaty define units of heredity. So there's nothing actually to stop us viewing DNA sequences in a database as being units of heredity, although that's not the way people have looked at them in the past. Nor do these agreements restrict the processes by which units of heredity are replicated. So there's nothing to stop us considering in vitro and in silico replication as being within scope of the agreements, to the extent that they're ultimately incorporated in genetic material that is commercialized. So that requires a change in perspective, looking at units of heredity and how they're replicated in a way that no one ever viewed them before. But if you take this perspective, it at least means you can consider fair and equitable benefit sharing from DSI without needing to amend the CBD or the treaty to bring DSI within scope. Because that would be a real problem. If we had to rewrite the CBD or the treaty, we'd be decades before we got there. But it does limit options for benefit sharing because it's essentially impossible to trace DSI from sequenced material to commercialized material. So that's the major difficulty here, getting a fair and equitable system of benefit sharing that doesn't rely on being able to trace where your information came from. Thirdly, the final issue, the issue that was recognized as a loophole many years ago, and Claudio already referred to it, that you can use materials from the multilateral system or other access and benefit sharing regime during the research phase to discover a desirable sequence and then find or obtain the desired sequence in an unregulated source and use it for product development. Hence, you can bypass benefit sharing obligations, which is clearly against the intent of the treaty and the CBD. And this really is a serious problem for fair and equitable sharing, benefit sharing. And that's really why we have to rethink ABS quite fundamentally in the era of DSI. Because it means there's a strong temptation to bypass benefit sharing obligations by seeking alternative resources from another country under a different access and benefit sharing agreement after you've done your research. I've given aromatic rice as an example because this is a highly valued and treasured trait that is carefully guarded by the countries that originally had it. So, for example, basmati rice in India and Pakistan and Thai jasmine rice have identical sequences of the major aroma gene. Aromatic varieties in several other countries also have identical sequences, and at one stage it was believed that all aromatic varieties had exactly the same sequence. But then we sequenced the genes in all aromatic varieties in the IRI gene bank and found quite a number of different sequences in many other countries with identical effect. Bangladesh, China, Indonesia, Japan, Laos, Madagascar, Malaysia, Myanmar, Philippines, and others. And we now know that in theory, many other alterations are possible. Because of the research that's been done, we know that non-aromatic varieties are non-aromatic because they have a gene that creates an enzyme that metabolizes the aromatic compound to a non-aromatic derivative. If you just knock out that gene, there's nothing to metabolize the aromatic compound, so its concentration just builds up and you get a perceptible aroma. And it doesn't matter how you knock out the gene. There are many different ways of knocking out genes. So now, armed with this knowledge, we can invent any new sequence that knocks out that gene and use gene editing to create an aromatic variety containing our gene. And we've driven a coach and horses through the politically very sensitive trait of aroma in rice. This is really quite a big problem for DSI and access and benefit sharing. So just a brief one slide on the implications that benefit sharing in the era of DSI can only be fair and equitable if it delinks payment obligations from the incorporation of DNA sequences into final products, and if it recognizes, reflects, and rewards the value contributed by DSI through upstream research. Now, CGIAR has been very proactive in pushing at achievable, effective solutions, which Mathieu and Claudio will address in the next presentation. But in the meantime, until they start, I'm happy to take questions and say thank you. Over to whoever's.
Thank you so much, Ray, for the excellent presentation, and Claudio as well. I think we will leave a few minutes for questions now. So before we move on to the next presentation, feel free to raise your hand or write your questions in the chat for our presenters. I will give people a few more minutes. If you want, you can stop sharing your screen, Rory.
Okay.
Up to you. Okay, we have a question from Ahmad. Ahmad, you have the floor.
Well, thank you very much. Okay, so my concern regarding access and benefit sharing is, which I would love to hear from the experts. As you can see, a lot of germplasm is being shared through proper channel between the research organizations, or between the labs, or between the scientists. And some of the germplasm which are not being shared through proper channels, but in different means or different ways, they have been transported from one country to another country, and they are being studied, being registered by the countries or the scientists that are not actually belonging from the origin of that germplasm. In that case, what do you think, uh, how you can protect the sovereignty of the germplasm of that actually original country, that from where the germplasm was originated actually? So whether DSI or the sequence information generated by the second country, if they are going to publish that sequence, they are going to claim the ownership of that germplasm? Or do you think there could be some ways the sovereignty of the germplasm, as we can see, the huge diversity can be is actually can be seen from the African countries or something like that. So how do you see how we can protect the sovereignty of the actual the germplasm from actual countries or host countries or the actual owners, we can say? Thank you very much.
What a big and important question. That's that question is really what drove the CBD to be negotiated, because everyone was really concerned about precisely that. The CBD solved many of the issues. One of the things it couldn't solve is the question of retroactivity. An awful lot of the material that you're talking about left the country of origin before the CBD came into force. And you can think of potatoes that left Latin America many hundred years ago. You can think of rubber that left its country of origin hundreds of years ago. You can't be completely retroactive in applying rules to everything that left the country before new rules came into place, which means we're able to consider your question properly for materials that have been removed from the country of origin since the CBD came into force. The CBD itself, you're reliant on countries to create their own legislation to maintain their sovereignty, to ensure that if other countries get access to their materials, they have a role in saying how benefits are to be shared. If you're talking about material that has been removed from the country of origin after the CBD came into force and against regulations, national and international regulations, then we have a big problem. And you should be notifying the countries where they came from. If you know of examples, there are procedures for notification under both the CBD and the plant treaty, if you know if there's been a breach of regulations. But it's, as I say, for the CBD, at least it's very limited. For the plant treaty, it partially overcomes that time limitation. For example, a lot of the materials in the CGIAR gene banks were removed from their country of origin before the CBT came into force. But even so, they have been put under the auspices of the plant treaty, and therefore the plant treaty regulations apply to them, even though they were taken out from the country of origin before CBD concepts came into force. So we have a degree of extra protection there for old materials. That's not going to get over the problem that there exist an awful lot of materials that are in private collections, removed legally from the country of origin when it was legal to take it. And I'm not sure there's a solution to that. Everyone is trying really hard to make sure we cover as much as possible. But at the end of the day, if a company or an individual has removed material before there was legislation, and you can't get at that person for patenting a sequence, if it's based on something that they've had for 50, 60, 70 years. Obviously, you question whether making a patent claim allows them to claim ownership of the material, and that there are some legal niceties there that it doesn't really mean they claim ownership of the material. And it's probably better if I hand over to Claudio to deal with those legally very complicated issues of what you're actually claiming when you get a patent. Claudio, do you want to talk more about that?
Thank you, Rory. Actually, what I'd like to add, so with respect to the patenting issue, we've published relatively recently a study on this particular topic of native traits, patent on native traits, and the evolving practice around patenting for that. So I won't spend more time here to discuss that issue because it's quite technical in a way, but I can post the link of the current version in the chat so that you can have easy access to it if you want to dig further. But perhaps I would like to complement by saying that a forward-looking answer to that question, because Rory has emphasized very much the retroactivity issues and materials that have been collected in the past. But going forward, it's also good to know that the Nagoya Protocol on Access and Benefit Sharing, which is a protocol to the Convention on Biological Diversity, has been adopted in 2010 at COP10, or the Conference of the Parties. And under the protocol, I mean, the reason why the protocol has been advocated for and negotiated was precisely to be able to enforce compliance in user countries, so not in the country of origin, but in other countries where the users are doing the research and development and bringing products to the market, so that the user country would have to say to the different companies that are operating in their territories and to check if they are complying with the legislation of the provider country, right? So that is known as the compliance pillar of the Nagoya Protocol. The European Union and its member states have been implementing it through a series of regulations. Other contracting parties have not yet implemented that. So it's work in progress. But in principle, for material, if your country is a country that has ratified the Nagoya Protocol and has adopted national access and benefit sharing legislation regulating that, then you can seek recourse under the mechanism of the Nagoya Protocol, under the compliance pillar, to seek enforcement of your legislation in another contracting parties to the protocol, where the user is undertaking the research and development activities or the commercialization of products. So there are mechanisms there to obtain compliance through enhanced cooperation mechanism that are established under the Nagoya Protocol. Having said that, I totally agree with what Rory said, that obviously it's easier to deal with these issues in a multilateral way, in a multilateral context, such as the multilateral system of the plant treaty, or also the multilateral mechanism under the Convention on Biological Diversity that we will discuss in a minute, because again, in that case, the boundary between things that were collected before or after entry into force of different instruments and so on gets bypassed by the fact that everything can be included by those who support and ratify these treaty mechanisms. And I'll post the link of the patent study in the chat. Thanks.
Yeah, perfect. Thank you. Very comprehensive answer and great insights on the question. Mike, then another linked question is that, okay, if we are talking about patenting and we are talking about CBD and Nagoya protocols, how do you see, for example, some of the, for example, developing countries, they are not able to sequence their genomes or their gene banks, and they are not able to register their germplasm or some of the key genes they are going to identify through genomics. In such cases, for example, in current era, still some of the countries, they are rich in genetic diversity, they have excellent resources, and they have potential germplasm that can revolutionize our agriculture, but they are unable to sequence them, they are unable to register them, they are unable to get a patent for certain genes or certain part of their genomics. In such cases, how do you see how the DSI or the treaty can protect their rights, can protect their sovereignty or the ownership? Thank you.
Who wants to take that?
You want to take that, Claudio or Mathieu? It's again another really thorny issue. You're picking up on precisely the reasons why DSI have become such a big issue. As Claudio said in his introduction, that there is this big gap in ability to sequence between developed and developing. And the next presentation will go into where we are in terms of the solutions. And I don't think we're there yet. It'll be some years probably before we're there, but we've got some partial solutions which will be presented, and maybe that's the best thing, is to see what the next presentation has to tell you.
Yes, just to complement on these very, very important questions. We as CGIAR are working in close cooperation with the Plant Treaty in delivering capacity building and capacity sharing for NARS, for all partners at national level, precisely to build capacity of countries, partners, and research organizations to do the sequencing, and also to capture the value that is added value that they are learning to generate progressively through these tools, and so on. Now we don't have the time to go into the details, but we are submitting 3 different reports, submissions to the governing body of the Plant Treaty that will take place in November this year, the 11th session of the governing body of the Plant Treaties. There's a wealth of details of the activities that we have been undertaking and on the plans going forward, including, of course, this series of webinars and in-person training later in November, and many other activities across CGIAR that we are trying to contribute to in this space. There's one of these reports that is specifically on capacity sharing for the use of digital sequence information. So if you don't mind, these reports are being finalized and submitted. we can circulate it to you as soon as they are ready through the mailing list that Alessandra has, and you can find more details there. And from there, of course, we can have a follow-up if you want to get involved more extensively in the activities that we plan going forward for the remaining part of this year and of course in 2026.
If I can just add something to your question, Ahmad, is that your question also refers also to the access to the information, and we'll be talking about database management and open access. And I think that it has been shown as well what is important is to make sure that the information is accessible whoever sequences the information and makes the DSI available. So there has been a study showing that even if some years ago, the sequencing was very expensive and it was mostly done in northern country, but those data were put public and the use of these sequences were also monitored over the years. And it was shown that overall it benefited to many countries. All the countries have accessed the material. So just to say that there is a complex relationship on the user on DSI. It's not black and white. So it benefits to many countries as well, and not only to the data generator. So just for information, I also copy in the chat this study if you want to have a look.
Thank you so much, colleague. Ahmad, I hope that answers your question.
Perfect.
And indeed, you will see more in the following presentation. So I think we can easily move on to the next presentation, since I saw that the question, or more the comment that was included in the chat, was already attended to by Rory and Mathieu. So thank you so much for that. I would say let's move to the next presentation. Claudio, if you want to do the honors, and then we can leave some more space for questions at the end. Thanks.
That's great. Thank you. Thank you so much, Alessandra. So, okay, let me share again if I'm capable of doing this again. Up. Right. Okay, perfect. So in this final presentation, we will look a little bit closer at the details and updates that concern the state of DSI rules and negotiations under the Convention on Biological Diversity and also under the Plant Treaty. I will start and then I'll pass the floor to Mathieu, who will talk about, in particular, the governance that relates to database management in relation to DSI, and then we will look finally at the Plant Treaty. So I would like to start by just highlighting the fact that under the if you are using DSI in your work, you should be cognizant of the fact that this DSI can be mixed together with other DSI in a global data set such as INSDC, which disregards the somewhat artificial distinction of competences between different treaty instruments and UN fora. But if you're working with plant genetic resources that are within the scope of the Plant Treaty, you will be, working in the context of the set of rules that have been established and that are, set out by the Standard Material Transfer Agreement that Rory mentioned. On the other hand, if you're working on so-called non-Annex 1 crops, that are the crops that are not within the scope of the multilateral system, then you would end up under the framework established by the Convention on Biological Diversity, the Nagoya Protocol, and nowadays the new multilateral mechanism that we are going to talk about in a minute. But it's important to understand also that if you're working, for example, on soil biodiversity, and interaction between microbes and perhaps pathogens, biological control agents. There are a number of other instruments, as we said, that could impact on your research activity, that at the level of use of the genetic materials, but this artificial distinction and fragmentation of international law with respect to the subject matter of genetic resources in different subsectors could also potentially be replicated with respect to digital sequence information. And that is very problematic, and that is why we have been arguing over and over in our participation in these treaty negotiations that we hope that the final outcome will lead to a system that is as harmonized as possible insofar as the governance of digital sequence information and benefit sharing is concerned across these instruments. So this is just to highlight this very important initial point of entry into our work. Now, turning our attention to the multilateral mechanism under the Convention on Biological Diversity. This mechanism was firstly adopted by COP15, so the 15th Conference of the Parties to the Convention on Biological Diversity, together and as a package with the Kunming-Montreal Global Biodiversity Framework, which is the framework that promotes action and implementation by member states to conserve and preserve biodiversity with a set of targets that have to be achieved by 2030 and with 4 overarching long-term goals up to 2050. Then the 16th Conference of the Parties to the Convention adopted another decision on DSI in November 2024 in Cali, whereby it operationalized, it provided for the technical details to operationalize this new multilateral mechanism on DSI. So the first practical element is that it applies only to digital sequence information. So the material genetic resources remain covered under the bilateral approach established by the Nagoya Protocol, okay? The other important element that we already pointed out is that DSI is not defined in the decision nor anywhere else. So the scope is not defining strictly the scope of DSI in a legal sense is not necessary to enable the functioning of this mechanism that does not require tracking and tracing of DSI into specific products. So this is the second element. With respect to monetary benefit sharing, the decision establishes that sectors and users in sectors that directly or indirectly benefit from the use of DSI in commercial activities should contribute at least 1% of profits or 0.1% of their revenue. They can choose between one or the other to the Cali Fund. That is a new fund that has been established under the Convention on Biological Diversity. So this is because the Cali Fund is indeed one element of the funding strategy to support implementation of the Kunming-Montreal Biodiversity Framework that I mentioned earlier, and to support and promote, the conservation effort, in particular of indigenous peoples and local communities in developing countries. So who are the ones who have to make these payments? It's not everybody, it's just entities or companies that exceed over the preceding 3 fiscal years the following thresholds. So they must exceed 2 thresholds out of 3. So the first is to have had at least 20 million in assets, US dollars. The second is to have generated a turnover of at least 50 million US dollars in sales. And the third threshold is to have yielded profits equal or above 5 million US dollars for the preceding 3 years of activity. Okay, so when these thresholds, when 2 out of 3 thresholds are met, then
in principle, the entities that have been using DSI would have to make payments based on the sales of products and processes, even licensing for processes under the convention. Interestingly, plant and animal breeding is explicitly listed in the indicative list of sector that is also annexed to this decision. So we are not, we are, catered for by this framework. With respect to the entities or company making the payments, they will be issued a certificate that will attest that they should be exempted from any additional payments linked to the use of DSI that are within the scope of the multilateral mechanism. So it's a sort of certificate of compliance, the equivalent under the Nagoya Protocol. So this is an annual certificate. So for the year in which a payment was made, they will have the certificate for that particular year, and so on. It's important also to note, however, that, as I mentioned in the introduction, countries are free to regulate bilaterally access to DSI under ABS contract. And so, in a way, having this certificate and having made this payment does not exempt from other additional obligations that might arise under national ABS law, such as, for instance, in the case of South Africa legislation and a few others. With respect to the issue of implementation, so how is this mechanism supposed to be implemented? What the decision says is that the parties, that is to say the member states, are invited to take measures to create incentives for users in their jurisdiction to contribute to the Cali Fund in line with the modalities that we have just described. What is important to emphasize here is that clearly the countries that have negotiated this agreement have stepped back from agreeing to requiring user in their countries to pay. So this formulation that we have here is a formulation that is non-legally binding. So these payments are, in a way, voluntary payments, right? It's not mandatory payments. And the way they will depend on the way in which different countries will take different measures to implement the decision. So at present, it is not clear if the payments will be made and how much will be generated by this mechanism, because there was an expectation indeed of developing countries that these would have been mandatory payments for all, which is not the case, as I just said. As far as we are directly concerned, entities operating public databases and public research academic institutions are not expected to make the payments, because we very likely are not going to meet the threshold requirements that I illustrated in the previous slide. However, our R&D partners, our licensees, users of data that we make available might be subject to these obligations, and therefore it's important to make them aware of the existence of such obligations. This nice infographic is just to, again, recall that non-commercial users are expected to make non-monetary contributions and benefit sharing in the form, for example, of scientific publication, open access to DSI, sharing of capacities, and so on. That is what we do, for example, as CGIAR, and what is illustrated in the report that I mentioned earlier. However, commercial users, on top of the non-monetary benefits, are also required or expected, at least, to make monetary contributions to the Cali Fund. As regards the management of DSI databases, there's a specific set of provisions that you can find in paragraph 10 of the decision implementing the multilateral mechanism that provides that the entities operating databases need to take a number of due diligence measures. This includes, for example, making information on the multilateral system available to those accessing the data, including the fact that they may be subject to the monetary benefit sharing payments should they, be commercial users. The databases should also informing those that submit data that they need to comply with national access to benefit sharing legislation. Furthermore, the entities submitting information into databases must provide information about the country of origin or the genetic resources from which the DSI are derived, as well as other metadata, including metadata on any relevant traditional knowledge that is associated to the genetic resources. With respect to data governance, access to DSI needs to be provided in compliance with the principles of fair care, trust, and other relevant applicable principles. And finally, the final requirement, which is a novelty and is very important to us, is that entities operating databases should request those submitting digital sequence information that they are, that such information is not subject to any restrictions that would prohibit its sharing. And that is precisely restriction, for example, deriving from the application of national ABS legislation. So there's this very nice infographic on the right that Mathieu has developed, and we also refer to a publication that he and his co-authors have published. And now I'll give the floor to Mathieu, please, to illustrate how these requirements can be implemented in practice.
Thank you, Claudio, and good morning, good afternoon, or good evening, everyone. So as you can imagine, there has been a lot of discussion on database management, and we have seen in the previous slide that databases, DSI databases and data access is really in the center of the picture, because this is a concern who access the database, who is using this information. And that's why it's important also to understand the dynamic of the existing databases, and reminding that there is no formal definition on DSI, but if you consider the different definitions that exist, we can say that in biology there are at least several thousands of databases for biology dependent on DSI information. And you can classify those databases in different types. So you have the ones that are large databases like INSDC that was mentioned before by Claudio, that includes NCBI, ENA, that are multi-species, non-specific, where people submit a lot of data. So this information is usually the main one that is being accessed, but you have as well a large ecosystem of secondary databases. So it can be thematic specific, but it can be dedicated to different crops, different plants. And all those databases create an ecosystem that is interconnected with exchange of information. So trying to regulate that is not an easy task. It's not like if you have only one institution, you have actually many countries where you have these databases, many institutions, and you have as well, as we can see on the diagram, you have also a lot of private databases, but for this one, as they are not public, it's difficult to know exactly how many there are, and they are not really regulated by the decision. But 2 years ago, we had a request from the Plant Treaty to make a kind of overview specific to plants, and we made this policy brief. So if you are interested to know a little bit more about this landscape, you can access the policy brief you have on the right on the slide. Next slide, please, Claudio. So if you attended my seminar on Wednesday, I mentioned that among this whole ecosystem of databases, the CGIAR is maintaining one, which is called GIGWA, and which is a system to facilitate the storage and the sharing of information from the CGIAR Genebanks. So in this system, this is where we can import SNP information, variant information, diversity information about the plant genetic resource that are conserved in all the gene banks of the CGIAR. And obviously, this system, as we are generating data and we are putting this data available online, we need to apply the full package of measure that was defined at the COP16 in the decision. Next slide, please. So here, I just want to give a few examples of what it implies, because as the system is, it is not fully compliant with the decision, which was adopted in November last year. But if we get back to the modalities that has been defined, the first modality in paragraph 10A is you need to make people user aware that there is a multilateral mechanism. So basically, what does it imply? If you have already on your system, like we do, a term of use, when people connect the website, there is a pop-up, they can access directly the term of use. You need to inform your user about the decision and the existence of the multilateral mechanism. So this would make it compliant with the modalities that are defined in 10A. Then switching to 10D is that we have to be consistent with open access. And so in our case, it's fine because the database is online and it's open access. But we need to take into consideration a number of principles that are fair, care, and trust, the ones that were mentioned in the decision, that are specific principles to facilitate how to find the information, how to access it, the interoperability and the reuse of information, so that would be fair. If you look at care, it's more about the data governance for indigenous data. So this type of information has to be treated separately and to take care about the origin of material. So most of the databases at the moment in the world are not really applying this type of principle, but there are a lot of database managers that are currently discussing those principles and see how it can be applicable to their systems. Next slide, please, Claudio. Looking at 10C, what was mentioned, the country of origin is some information that is very important and must be specified when you are submitting your data. So here again, in our system, there is a way to submit metadata. And what is important is to make sure that when those metadata are submitted by user, we make mandatory the country of origin, as well as a unique identifier of the accession if it exists in Genebanks. So if you don't know about the country of origin, you can still put unknown, but as much information you have on your material, you should make it available, as well as traditional knowledge. So it is possible to create additional column in the system and to put any information that would make it linked to traditional knowledge associated to your genetic resources. Next slide, please. And in terms of data submission, so what you see here is basically the interface you have to fill in when you submit your data set. You need to inform the data uploaders that they need to certify that they have the permission to submit this DSI, so that there is no regulation that would prevent further sharing of the information as open access. And for this will be a big change in most of the databases because it does not exist. But here, it should not be too complex to implement. It would be basically some kind of checkbox where you ask user just to certify they have the necessary permission. So this is quite easy to implement, and we expect that people will do it in most of the databases to respect the modalities of the decision. So I think that was my last slide, and I hope it is useful because at the beginning you mentioned that some of you are managing information or may have databases you want to share. So those are examples of how you can apply change in your system if you want to comply with the modalities of the COP16 and the decision. Thank you very much.
Thank you so much, Mathieu. I think it's always great to see these practical ways to implement the CBD decision in a very visual and practical way. So I'm cognizant of time, and okay, let me skip this one. I just wanted to flag to provide you with an update on the plant treaties multilateral system enhancement process, which is currently ongoing. Just in a few minutes, and apologies if I don't cover it to the extent and with the granularity that we might have wished, but just to say that again, there's a process that started in 2013 to enhance the multilateral system of the plant treaty. That was triggered primarily by the fact that the payments that were going to the benefit sharing fund were mostly voluntary payments by member states, and there's less than a handful of companies that have made payments under the mandatory benefit sharing modality of the standard material transfer agreement. So there's an expectation that on the one hand, the payment basis will be more predictable and increased through this revision process. And on the other hand, that also the plant treaties multilateral system would be expanded to additional crops and forage species, right? So this process has been ongoing for more than 10 years now. As you can see, it is expected to be finalized at the next meeting of the governing body in November 2025 in Lima, Peru, yes. And because of that, again, we will have perhaps a full module in the in-person training later in December when you can provide you with additional detail about this. But just to say that the main challenge in the context of these negotiations, which are ongoing, is to layer on, again, obligations for DSI on a system that was initially designed for using and accessing just the physical materials. So the negotiations are taking place recently based on what is known as the June 2019 draft package of measures, which includes a draft amendments of Annex 1 expanding the scope of the multilateral system, a draft revised standard material transfer agreement, as well as a resolution on implementation where it is clarified also how the extent to which benefit sharing from DSI is catered for under these new payment mechanisms that are established under the revised standard material transfer agreement. With respect to monetary benefit sharing, nothing has been agreed yet, obviously. As I said, this is an ongoing process, but we are heading towards a system that will be based on two possible payment options under the SMTA that the recipient can choose from. One is a subscription option, whereby subscriber will subscribe for a minimum period of 10 years. During this period, they can access any PGRFA from the multilateral system, the CGIAR, whoever makes materials available in the multilateral system, and they will start making payments from the year in which they have subscribed based on payments that are calculated on the sales of any PGRFA product that they have in their portfolio. So this is different from the current situation, of course, where you need physical incorporation of materials in a new plant variety that you commercialize, as Rouard explained earlier. So the rate of payments would be very low because, of course, the payment basis will be potentially very large for subscribers. So very low rate, access to everything, but predictable income for the benefit sharing fund because the payments are due from the time of subscription. The second option is known as the single access option, that is very similar to what we currently have under the SMTA. So we still have an incorporation, physical incorporation requirement into a new plant variety that is being commercialized. The payment will be based on a higher percentage. The percentage have not been fully discussed by parties yet, so it will be for the governing bodies to decide what these rates will be. But we know there's an indicative rate that is what is the current one, that is 0.77 for products that are available without restrictions. So products that are covered either by a patent or by a restrictive licensing agreement that does not allow the user to do research and development with the material, right? And then probably an intermediary rate that will be higher than higher than the subscription option rate, but lower than the single access for products available without restrictions, for products that are available for further research and breeding, such as, for example, varieties that are commercialized under plant variety protection certificates or that are in the public domain. So for DSI, several options have been considered, but none has really caught on so far. And this leads to final consideration on what is the relationship between the enhanced multilateral system and the multilateral mechanism under the Convention on Biological Diversity. And here, in our view, it is clear that, for example, for subscribers, because when they make payments under a subscription system, those payments cover a broad range of broad portfolio of products that they have, and that the trigger for those payments disregards the physical incorporation of materials into these products, then these should cater also for any benefit sharing that is due for the use of digital sequence information. So they should be exempted from any additional payment under the multilateral mechanism. And this is something that is captured, as I said earlier, under the draft resolution as part of the package. On the other hand, for users that choose the single access option, if no benefit sharing for DSI is ultimately structured into the package, as it is the case right now, it seems logical that then if these users use digital sequence information from PGRFA, they will have to make payments also under the multilateral mechanism, under the convention, if the triggers that we have seen in the first part of the presentation apply to them. So this is really in a nutshell. We know there's a lot to take in and there's not enough level of granularity, but I thank you very much for your attention. And also I would like to direct you to our resource page on digital sequence information and plant genetic resources, which includes link to many webinars that we have undertaken and that we update regularly as this negotiation progress, to our scientific publication and to other awareness raising materials that could be of your interest. So with that, I thank you very much and look forward to any questions that you might have.
Thank you so much, Claudio and Mathieu, for this extensive presentation. You're getting quite a lot of positive reaction from the group, which is great. So I will now leave the floor to our participants to see what they think and if they have any questions regarding the presentations. Feel free to raise your hand or write in the chat, however you prefer. And please note that for those of you asking for the presentations, all presentations will be shared together with the recordings at the end of the webinar series. So most likely next week, to give us some time to collect everything. I see no question. Oh, Ahmad, please go ahead. You have the floor.
Thank you very much. Another a point was coming in my mind, and I would be happy to hear from the experts. as sequencing technologies are progressing day by day, and one group or one country or one scientist, if they have sequenced one genotype in 2025, and then with the passage of time, if they sequence the same genotype after a couple of months or couple of years, uh, we can see there could be some natural variations in that genotype, and based on the natural variations there could be some differences in the sequence. Then in that case, how the treaty will look at that germplasm or the genotype, whether they will consider it as a same or the different genotype, how they will, I mean, they will consider as an entry point of view. When one organization is registering the same genotype with the same metadata, but with a different with the different sequence information, in such cases, how the treaty will deal with such entries, or the CGIAR, for example, if you have the database like what it is, GIGWA. Okay, when they are going to submit to GIGWA, they have the same metadata, but the sequence information is different. Then how you are going to deal with that?
I can try to reply your question. Well, it would, if the sequencing is made on the same genotype that has evolved over time, it would remain in terms of metadata the same, which is normal. And actually, this is what already happened in Genebanks. If we consider clonal crops, for instance, I've been working for a very long time with banana at the International Genebank, and you multiply the material, and some modification can happen, and the people that are receiving material year after may have accessions that are actually some modification compared to the one that was conserved or distributed some years ago. And even sometimes you can sequence with two different methods, more or less at the same time, and you can find variation. Even using different software in bioinformatics, sometimes you can have some variation. So I don't think it would make a big difference in terms of origin. What is important is the origin of your material, that your material is properly identified, and even if there has been some variation in the genome, it would remain. Considering Genebanks, it would remain the same. It should be treated the same.
Add something to that?
Coverage. Yes.
This is more a scientific question than a policy question, I think. As Mathieu has said, even in the clonal crops, you can get variation just sometimes for software reasons, sometimes for mutations. It's a much bigger issue for the seed crops, because there, as you're implying, one accession is normally, even for an inbreeding crop like rice or wheat, one accession is actually a population of many different genotypes. And CGIAR has put a lot of work into trying to establish scientific procedures, best practices for sequencing materials. And for the seed crops, not so much for the clonal crops because it's not such a problem, but for the seed crops, whenever we sequence, we would select as far as possible a pure line out of the original accession, so that as far as possible, having sequenced it once, it's not going to change in the future. Of course, you can do several generations of selfing and still you end up with some residual diversity, so it's not a complete solution. But definitely, it's a scientific matter. Do everything you can to make sure that you have a pure line that accurately represents what you have sequenced. And when you publish it, don't say the metadata are the original accession, because the original accession is a mixture of genotypes. When you publish it, you say this is a single line selection from your original pure material. And in that case, we can more readily trace it to the specific single plant selection that you've made. As I say, this is not really a matter for the treaty. This is a matter for the scientists to operate good practice to make sure that the genotyping is fixed. Because if you genotype a mixture of accessions, sorry, a mixture of genotypes in one accession, then when you come to phenotype that accession, you will probably find that what you're phenotyping is not the same genotype as you have sequenced. So all your phenotype-genotype associations break down. You have to do that pure line selection before you start to sequence, and especially if you're going to phenotype that material, to look for phenotype-genotype associations. It's really a scientific issue, but it's a really important one.
Thank you very much. That's great to hear. Just from information, another question: is GIGWA accepting the sequence data from the other parties other than the CGIAR banks?
Well, if your question is to know if you can use the system, I mean, it's an open source software that is actually developed at Rouaraidh in France. So anyone has a possibility to download it and install it and use it. So we have put in place specific instances for CGIAR Genebanks, but it doesn't prevent anyone to adopt the system, a different instance, and use it for their own needs. So if it was the main idea of your question.
Yeah, it is just operating just like an NCBI, Phytosome, or other databases, or differently?
No, well, it's not centralized. It's you can, for instance, download it and put it on your computer and manage your own data, but you can put it on a server and propose an access to a group of people. You can replicate some instances in different places. So it's very flexible. It really depends on your use. But in our case, we have tried to put some server with a common access for the different Genebanks, but it can be quite different according to different organizations who would like to install it.
Great. Thank you. Thank you very much.
You're welcome.
Thank you, Mathieu and colleagues, and Ahmad, for that question. I hope you got the answers you needed. We have a few more minutes, a couple minutes to get additional questions, if anyone has any. Feel free to raise your hand or write in the chat. Otherwise, I will leave the floor to my colleagues for the quiz and end of webinar survey. No questions? Okay, so then Obina, do you want to take over? I don't know if Marie-Noëlle has to say a few words prior, or if you want to launch the quiz. In the meantime, I would like to thank once again our wonderful speakers. They may stick around if you have some questions. Otherwise, you will receive their presentations, and their email are at the end of that presentation. So if you have any questions, you can also reach out to them. Thank you so much, colleagues. Obina, you have the floor, or Marie-Noëlle, whoever would like to introduce the quiz.
Hello, everyone. I will be walking you through a very quick interactive quiz session. Just a minute to have my
Are you trying to share your screen, Albina?
Yes, just a minute.
Okay.
Okay, so we'll be walking through a very short and interactive quiz just to assess the different information you've gathered over the last 10 sessions. So quickly, I would want you to scan the barcode. Please scan the barcode. If you cannot scan the barcode, I will be sharing the link to the survey.
Please send us a thumbs up if it works for you, the QR code, so we can check.
Okay. So can I get any thumbs up?
I saw a couple. That's comforting. Let's see. Yeah,
Okay.
there's more.
Great. So once you've scanned the barcode or you've used the link within the chat box, feel free to enter your mails, very important, and let's know you a little. Let's know your role and institution. We're just going to give 20 seconds for that, and then we will move to the questions. So again, if you've done that, let's have some thumbs up. Can I get any thumbs up? Okay, I take it you've done that. So we'll quickly move to the next first question for the day. I'm going to help you read the question and you would provide just the answer on the link. So the first question is, what was the primary strategic advantage of the Latin American community of practice? This was treated on the first day of the session. So can we have some response? Please indicate in the chat if you've provided a response to that. Okay, moving forward, we're going to see the next question. According to the presentations from CIP, commercial DNA extraction kits are always the superior choice over the CTAB method for gene bank work because they yield higher quantities of DNA and are effective for all plant species. including those in high mucilage and polyphenols. So this was a question from the second day. So if you were there, you're allowed to choose true or false. It would be lovely to have some reactions. If you're actually answering, can I either see your hands, your thumbs up, or put in a notification on the chat? Okay, I think I get something from Tina. Well done, Tina. Okay, moving forward, the third question, I will need you to match the DNA quality assessment ratio with what it primarily indicates. So all you have to do is match the 1 to 4 across the A to D. So we have the 260/280 ratio, the 260/230 ratio, the high sequencing depth, the low sequencing depth. Feel free to match its correspondence on the chart, on the form. Again, if you've done that, can I see some reactions? Anyone? Okay, moving forward to question number 4, just 5 more questions to go. A participant wants to use the Sanger sequencing to verify a cultivar. Can you place the steps of the Sanger sequencing for cultivar verification in the correct chronological order? That is to say, assign the number 1 to the first step, number 2, and so on. So we have options A to E. Which option do you think comes first, and which option do you think comes last? Again, if you've answered the question, I would love to see some reactions or either something on the chart so we can move on. Moving on to the 5th question, we have 4 more questions to go. This was on the last day of the first week. So based on ICRISAT's empirical research using genomic data, must a gene bank manager always regenerate a minimum of 458 plants for every crop accession to preserve rare alleles? So your options are yes, there is a universal rule. This is a universal rule for all seed crops. And then the other option is no. The optimal population size is crop specific based on its reproductive biology. Again, if you've answered the question, can I see some reactions? Moving forward, question 6. What is the fundamental biological principle that makes cryptopreservation at 196 degrees a viable long-term storage method? So you could choose from A to D. I already see that some people have done answering the question. Some people are faster than me. That's great. Okay. So the first is it completely sterilizes the plant material, eliminating all viruses and pathogens. B, it dramatically speeds up the plant's metabolism to promote rapid growth upon thawing. C, it achieves vitrification, a glass-like state that prevents the formation of lethal intracellular ice crystals. And D, it uses cryoprotectants to permanently alter the plant's DNA for better stress tolerance. Moving forward to the seventh question, 2 more to go. For the recalcitrant seeds like mango or avocado, the best practice for cryopreservation is to dry the whole seed to a low moisture content and then plunge it directly into liquid nitrogen. True or false? Okay. If you've answered that, we'll quickly jump to the 8th question. The open source tool, GigWar, is powerful for gene bankers because it primarily allows them to A, automate the physical process of DNA extraction in the lab, perform complex command line bioinformatic analysis without any training. C, interactively explore, filter, visualize, and export subsets of large genotyping datasets through a user-friendly web interface. And D, generate synthetic polyploid plants for breeding programs. So we already have 3 responses coming in. That's great. And so to the last question for today. The last question says, you have a VCF file from a genotyping experiment. You calculate the ratio of allele depth to total depth for the dominant allele at numerous SNP sites. What ploidy level is most likely indicated by the following average ratios? So all you have to do is match 1 to 3 to A, B, C. And this rounds up the questions for today. So if you are at the last question, please submit. And thank you all so much for participating.
Wow. Thank you very much, Clinton, for this nice exercise. Thank you so much. Wow. So you will analyze the result and share with us later on. So, dear participants, we are at the end of the 2 weeks webinar. And on behalf of Vanya, who is not here today, who is not with us because she's attending other meetings. Vanya is actually, somebody speaking, is actually the CGIAR, the director of the CGIAR Genebanks. I would like really to sincerely thank all the speakers who actually make this webinar happen, and all the participants, all the organizers for their time, their insight, and the active engagement. Please, I would like everybody to join me so that we can say thank you to all the speakers, to yourself, the participant, because you were able to join us at each of the webinars and to the organizer of this webinar. Together, we have explored the use of the advanced molecular techniques in the gene bank. Oh, I think Louise wants to speak. Okay, no, maybe it was just an error. Together, we have explored the use of advanced molecular techniques in the gene bank, the cryopreservation of the clonal crop, and also on the recalcitrant seed, the ploidy analysis, the GSI and the related issue with the multilateral system, also the CBD. This happened through rich discussion, diverse perspective, and thoughtful exchange that have deepened our collective understanding. I don't think today will be the end of this conversation. I think it's just a beginning, a starting. We will continue this dialogue through different means. I really encourage each of you to take forward the lesson, the connection, and the idea that have emerged from these days to your institutions, policy, and your daily work. Please implement it. Help us to translate all this knowledge into key actions to change the way you work, to change the way you work in your institution. So really, I'm so happy, and I'm really declare that this webinar is closed now. And then thank you all for your time and the time you have been devoted to this webinar. Thank you so much, and see you in the future.
Thank you, Marie-Noëlle. Could we take a picture maybe?
Great idea.
What do you think?
Yes,
so
great idea.
If you could all start turning on your camera, I'm giving you permission to do that. So then we can take a picture just as a souvenir. So please, if you're able to turn on the camera.
Maybe you should enable the camera,
Yeah,
I don't know.
I am enabling to everyone, but I don't see anyone turning it on. Perhaps people are shy. There we go, some people. That's nice. Hello. Nice to see so many faces. Abdel Karim, do you have a question? You have your hand up. No? Okay. Okay, I think all of you should have permission to turn on your camera. So those of you that want, please turn it on so we can take a picture.
You see the camera, they cannot open the camera, I don't know.
Yeah, I don't know why. I'm giving permission to everyone. But it may be a personal issue. Okay, so thank you, everyone. I'm taking a picture now. Great. Thanks so much. So yeah, this was an honor, and I hope there will be more instances of this in the future. Marie-Noëlle, thank you so much for also giving me this responsibility. I appreciate it. You're muted. Or no, sorry.
Thank you to all our speakers. Thank you to the organizing team, Clinton, Fa, Alexandra. Thank you so much to the participants for your time and for sharing all the nice discussion with us. Thank you. Bye-bye.
Thank you so much. Bye-bye.
Thank you. Bye.
Bye-bye.